Hey Mustangs, in this video we're going to be taking a look at the second stage of photosynthesis, which is the Calvin cycle. Now we already looked at the first stage, which was light-dependent reactions, and we saw that plants are able to convert solar energy into chemical energy in the form of ATP and NADPH. Now the Calvin cycle is dependent directly upon the light-dependent reactions because it can't happen without the ATP and the NADPH. So it needs the light-dependent reactions to happen first. So there is no Calvin cycle without the light-dependent reactions. Now, even though the Calvin cycle depends heavily upon the light-dependent reactions, my favorite part of photosynthesis is actually the Calvin cycle. Because from the human perspective, it is plants creating life out of thin air. You know, we can't see, we can't touch the air, so to us it's like, there's really nothing there, even though there's molecules all around us. And specifically the carbon dioxide that the plants are pulling out of the air in order to make stuff. So that's why the Calvin cycle is my favorite part. Seemingly, from the human perspective, it's making life, making molecules, making those molecules that other living things, and not just the plant, but other living things depend upon out of thin air. So let's go ahead and take a look at the Calvin cycle. So the purpose, why does the Calvin cycle happen? The Calvin cycle is going to take the ATP and the NADPH, and it's going to use that to reduce carbon dioxide to make the sugar G3P. So there's a few key things to hit upon here for this purpose. The fact that it's reducing. So reduction means adding hydrogens, which is adding electrons, which is therefore adding energy. The second important part of this purpose that we need to know is the G3P. You might be thinking, wait, I thought the Calvin cycle made glucose. Well, we learned that the first time we learned about photosynthesis in 9th grade and all that stuff, but in reality, the Calvin cycle and photosynthesis as a whole makes a different sugar called G3P. That G3P can be used to make glucose, but it's actually not what's made at the end of photosynthesis. It's not what's made at the end of the Calvin cycle. It's the G3P. So, summing it up, Calvin cycle is going to reduce carbon dioxide, add hydrogens to it in order to make the sugar G3P. When you look at carbon dioxide has very little energy in it. If you look at glucose, which we want to make after we make the G3P, we want to make glucose from that. Glucose has a lot of energy. It's highly reduced. It had a lot of hydrogens, therefore energy added to it. So when we look at the Calvin cycle, we can add several terms that we've learned in the past to help us understand what's going on here. So the Calvin cycle is going to be a series of steps that adds energy in order to make a sugar that has a lot of energy in it. So we can say that it's an anabolic process, so it's actually building something. So anabolic, building, versus catabolic, breaking apart. So it's anabolic, and it's also endergonic. You have to put in energy, and when you're putting in energy, that energy also gets stored as well. So it takes energy in order for photosynthesis to occur, because you're putting energy into the sugars that you're making. So it's endergonic. The Calvin cycle takes place in the stroma versus the light-dependent reactions, which took place in the thylakoid and thylakoid membranes. So light-dependent reactions there, Calvin cycle inside the stroma. There are 3 phases to the Calvin cycle. So when we take a look, it's going to look complex, but it's actually really easy. You just got to break down phase by phase in order to really understand what's going on. So the 3 phases of the Calvin cycle are carbon fixation, reduction, and then finally your regeneration phase. So it is a cycle, which means, take you back to another cycle here to help you understand this, the Krebs cycle. So remember, the Krebs cycle started with oxaloacetate, and then we added stuff to that, went around a cycle, things get broken apart, series of steps, so we go back to oxaloacetate at the end. Here, same thing's going to happen, except it's not going to be oxaloacetate, it's going to be the RUBP. So we're going to see what RUBP is in a moment, but that's going to be our starting molecule. This time adding carbon dioxide, going around a cycle and making RUBP back at the end, so the cycle keeps on going. Alright, so here is our complex looking Calvin cycle. So when you look at it, we can see our phases here. We have our carbon fixation phase, our reduction phase, and then finally our regeneration phase back to RUBP, which is a molecule that we started with. So let's take a look first at the carbon fixation phase. So here you're going to take 3 carbon dioxide molecules and add it to 3 RUBP using a very, very important enzyme. And in fact, it's the king of enzymes, king of proteins, because it's probably the most abundant protein on the planet. So it's called Rubisco for short. It's a more complex name, and even this complex name that I have here is shortened up. RUBP oxygenase carboxylase. The ASE tells us that it's an enzyme. Oxygenase, it can involve oxygen or it can involve carbon dioxide. And we're going to see that's actually going to be a problem later on, so keep that in mind. So here, our Rubisco enzyme is going to be used to take our RUBP and carbon dioxide and add the carbon dioxide to the RUBP. And that's what the Rubisco enzyme is going to do. And that's going to be carbon fixation. So carbon fixation is simply just adding carbon dioxide to organic molecules. Anytime you're incorporating carbon dioxide into organic molecules, that is our carbon fixation that we see happening here. Now, when this happens, it's going to make a 6-carbon intermediate. So we see 1, 2, 3, 4, 5, 6. And that's going to be really short-lived. So that 6-carbon intermediate is not the PGA that we're looking at here. It's just simply a short-lived 6-carbon intermediate. So it's going to look something like this. Okay? If you take 3 carbon dioxides and 3 RUBPs, here's our 6-carbon intermediates. You can see the skeleton of the RUBP there, and here's where our carbon dioxide comes in. This 6-carbon intermediate is going to split right away because it's very unstable, and kind of just picture it cleaving right there. And if it does, you'll have 3 carbons and a phosphate here, 3 carbons and a phosphate there, and that is our PGA. Okay, so that is our PGA right there. That is the end of carbon fixation. We've added carbon dioxide into organic molecules here. All right, now we have our reduction phase. So a reduction phase is going to involve taking 6 ATP, 6 NADPH, and using it to reduce PGA. So add hydrogens onto PGA. And when we do that, that is when we make our G3P. So if you take a look here at this image, this is what PGA looks like if we were to see the formula written out. So here's how we're representing PGA here with our circles and our phosphates. But here's PGA, and this is what you want to pay attention to at the end here. Okay, so in order to reduce PGA to G3P, first thing that has to happen is to phosphorylate it. So that's where the 6 ATP come in. So 6 ATP donate a phosphate, turning them into ADP, and they phosphorylate the PGA. So now you can see they all have phosphate added onto the ends here. But we haven't reduced it yet. So then the NADPH come in, and they're going to donate their hydrogens here and replace the phosphate that was just added on by the ATP. So all of these phosphates that were just added on will be replaced, and you'll go back to this. And you might be thinking, wait, that looks just like the PGA, but that's why I have these images here. So here was the PGA. Looking at that, that's where the phosphate got added on from the ATP. And then when we reduced it with the NADPH, we replaced that phosphate with the hydrogen there. So here's our G3P finally right here. Now we get into the regeneration phase. Okay, so we've made 6 G3P, but all 6 of those G3P aren't going to leave the cycle. Okay, in fact, only 1 G3P leaves the cycle. The other 5 G3P are going to be used in the regeneration phase in order to make the RUBP again, to start the cycle all over again. So only 1 G3P actually leaves. The other ones are going to be used to regenerate RUBP again. Okay, now it's going to be a series of steps. You don't have to know the steps, but a series of steps are going to occur, and it's going to involve using 3 more ATP in order to get back to the RUBP. Okay, this is a really cool one that I got actually from another teacher who obviously put a lot into this slide here. So here's our 3 RUBP. Here is our 3 carbon dioxide molecules. Okay, mix our 6 carbon intermediate. And that's our carbon fixation. Okay, there's our PGA. Okay, so the 6 carbon splits into the PGA. And then we add 6 ATP to phosphorylate the PGA. And then after phosphorylating it, then we reduce it by adding the 6 NADPH. And that gets us into our reduction and our formation of the G3P. We make 6 G3P. Only one of them is actually going to leave. So only one of them is a product here. The other 5 are going to be going back into the regeneration phase with 3 ATP in order to make RUBP again. All right, so the numbers. What goes in? We have 3 carbon dioxides that go in, 3 RUBP, 9 ATP total, and 6 NADPH. So there's a lot of energy going in here. This is what makes it end organic. Remember, ATP is our energy currency, so we're putting in a lot of energy to make our G3P. What comes out? 1 G3P, 9 ADP plus phosphates, 6 NADP pluses, and remember these are going to go back from the stroma here back to the thylakoid to be used again and again, and then 3 RUBP. Okay, so we regenerate back to the RUBP that we started with. Now, what would it take to make glucose? So if you remember our equation for photosynthesis, 6 CO2, 6 H2O plus gets you glucose and 6 oxygen. And don't forget the light, so I'm missing light here, but light energy as involved can't happen without light-dependent reactions. You'd have to do this twice. Okay, so you'd multiply everything times 2 in order to make your glucose, because you could take 2 G3Ps in order to make 1 glucose. Because you can see G3P has 3 carbon dioxides, glucose has 6, so 3 plus 3 gets you your glucose there. So basically you multiply this times 2. All right, so what happens to the G3P? It can be used to make carbohydrates such as glucose, which can then be used by the plant in cellular respiration. So remember, plants have mitochondria. They go through cellular respiration too. People always think that plants just do photosynthesis and that's it, and that's the end of it. Now, they do photosynthesis and then they send their glucose over to the mitochondria to go through cellular respiration to make more ATP. Second thing, it can be used to make sucrose. So sucrose is how sugars kind of get around through the plant. So sucrose is a disaccharide, and it transports through the plant, through the different veins and different transport areas. And once it gets to different cells throughout the plant, that sucrose can be broken apart, rearranged in order to make your proteins, lipids, and other products needed for the plant. It can be used to make glucose, so you could take that G3P, make glucose, and then chain it together in order to make cellulose, which all plant walls are made out of. So this is huge right here. You can't have a plant cell without a cell wall. So you take the G3P, make glucose, chain it together, and now you have cellulose, making your cell walls. Fourth thing that can happen, it can be just put into storage. So if you remember starch, starch is very similar to cellulose with a slight difference. See if you remember what it is, okay? You chain glucose together to make starch, and then that can be stored in different parts of the plant as well. So that's storage. And if a plant ever needs it, maybe photosynthesis isn't running right, which we'll see in photo respiration, what can happen. Plants can get the starch and break it apart down into glucose and use it for stuff like cellular respiration. So taking G3P, making glucose, and then chaining it together to make starch for storage for future needs. And then finally, it can be made into sucrose, glucose, cellulose, starch, proteins, lipids, which are then eaten by heterotrophs. So either part of the plant or the whole plant could actually be eaten by a heterotroph, and that stuff gets passed on. So now what the plant made is becoming part of other living things. All right, so summary of photosynthesis. Here is our equation, and you should be able to answer the following questions. So where did CO2 come from? Where did CO2 go? Where did H2O come from? Where did H2O go? Where did energy come from? What's the energy used for? What will the glucose be used for? Where did oxygen come from? Where will oxygen go? And finally, you should be able to list other things that are involved, such as ATP and ADPH and all that stuff. All right. And again, if you notice down in the corner, it says Foglia. So this is some cool stuff that I found from her. The thing about photosynthesis, it is the process that makes life possible on Earth. So all life depends on photosynthesis. If it doesn't happen, life is not possible on our planet. So it's probably the most important process on our planet. Each year, it captures 121 billion tons of carbon dioxide, and then it synthesizes 160 billion tons of carbohydrates. And of course, heterotrophs depend upon this. So plants are the ones that can do this, but all life depends upon it, because when plants get eaten, all that stuff gets passed along, and then heterotrophs can incorporate it into their bodies and get energy out of it in cellular respiration. All right, and then this was really cool. I really like this one that Foglia had created here. So all the solid material of every plant was built by sunlight out of thin air. All the solid material of every animal was built from plant material. So every animal was built from plant material. Therefore, all the plants cats, dogs, elephants, and people are really particles of air woven together by strands of sunlight. Very poetic right there. I love that. So there's that whole out of thin air thing.